Okay, we don't have much of time to watch our soccer game, so let me speed up. So I'm Shin Choi, founder and the CEO of the Elegslab. So we are saving our friends by, with the real bio world big data and then AI. Okay. Yeah, so millions of rats and mouse are sacrificed, and then 10,000, 100,000, dogs and rabbits are killed by, killed in the laboratory for the drug discovery and drug development. But about 90% of these drug candidates have failed in the phase 1 clinical trials. Even these animals are mammals, and the similarity is very close to the human being, but the number of data is still, small. So our strategy is like this. So even though, overcome this, problem, so many research groups are developing the in silico-based, AI, and then animal experiment alternatives like organoids or organ-on-a-chip. But there are no real bio-world data in this AI, and then there are no standardized big data in these animal experiment alternatives. So we are suggesting bio-experimental data is a big data, massive big data, and then we can track the animals using the AI model and our automation system. So these, three, approaches can guarantee the increase the success ratio of the clinical trial. So we are developing and manufacturing these boxes. So these equipment is collecting the images and movie clips from these, small tiny chips. It's the size of the thumbnail. When you look at the width of the 1 centimeter, and then there are 20 or 30 the small tiny worms are loaded, and then the camera, observing this the animal behavior or the movement or the response, and then they collect tons of data, and then we can track this individual worms using the vision AI. So the vision AI enables us, track the animals and then extract the bunch of the data. And then after collecting this data, behavior data, and then we can also collect the molecular, the cellular data after, entrap these animals in the channels. And then these tons of data are studied by the reasoning AI. So we are developing this reasoning AI, and this AI predicts, which candidates are best. So out of the, the 100 or thousands of the candidate molecules, and then this reasoning AI also predict the toxicity or the mechanism or the MOA of this specific, the lead compound of the candidate. And then this, small tiny worms is beautiful, the model. So we can establish more than 2,000, human disease model, and then these animals, I think it's broken. Is really good at for the cancer and metabolic disorders, and then toxicity or the aging or the neurodegenerative disease. So we have the triangular revenue structure. So we can sell these equipment, the boxes. These boxes can enable us to save time and labor and cost. And then we also are selling this biochip kit, is disposable things. And then these boxes and then these chip kits are operated by the AI. So these AIs are sold by subscription-based sales. So this boxes equipment has a size of market about $15 billion, and then this disposable chip kit has a $60 billion size, and then the AI, the subscription has a $10 billion the market size. So we're gonna ride big wave of this global supply chain through this the global, international, the big supply chains like the Jackson Laboratory or Thermo Fisher. So we have complete the POC with the Ujong Bio. It's a CRO, non-clinical trial CRO company in South Korea. And then through the open innovation sponsored by the government. And then we are contacting with the other, the CRO, the companies. The primary customers are these, CRO companies. And the secondary, our partner or the customers are gonna be the global big pharmas like Johnson & Johnson, AbbVie. So we can do this, non-clinical trial testing in-house using these, boxes and chip kits. And then we can also sell these, boxes and kits to this global big pharma, and then they can do this, automation and then AI-driven the non-clinical trial in that companies. So these are our C-level. So I'm the person more like the drug development and drug production guy, and then the CTO, the Dr. Joe, is a professional at developing the AI model and then fabricating the chip kits things. And our CSO is very specialized at the business development. So our company is located in Gwangju and Yongin near the Seoul area, and then you can scan me this LinkedIn the QR code. Okay, thank you. I'm happy to take any question from the floor.
You go back to the picture, the other one. Oh, it's very similar to this.
This one?
Yeah, that one.
Yeah.
How did you resemble?
Resemble.
How you can say that this study, right, with the worm matches the study in an animal in this case?
Oh, okay. Yeah. So there are many, the studies. The comparable study between the C. elegans and then the mouse model, like this. So these are two representative cases of the drug development through the C. elegans to the mouse model. The first case is the metabolic disease targeting drugs. So when you look at here, so there are about 10,000 candidate, the genes. Is about the gene therapy things. So they screened out like this, 44 the candidate lead compounds, lead genes from the C. elegans study, and then these, the lead compounds are also tested using the mouse models. So and then they selected one the most powerful the candidate, and they are doing the clinical trial. So this study is a really good example of this, C. elegans and mouse, the correlation, the study. And the second one is about the Lou Gehrig's disease, ALS. So it's about the small molecule, the screening. So they screened out, about 4,000 drugs candidates, and they found, really good compounds, 11 hits. And then these are tested using the mouse model, and then they are now doing the clinical trial. So about, there are really good review papers, meta-analysis through the 500 papers. It said there are over the 90% correlation between the C. elegans and the mouse in terms of the results of human disease things. Yeah, you're welcome.